In this video, we continue our discussion of proteomics, which is a really valuable experimental area for determining the differences between proteins in treatment samples versus controls. For example, if one were looking to establish particular proteins that are related to a disease state, one could collect protein samples from healthy individuals relative to diseased individuals and compare the protein profiles between those so-called treatment diseased individuals versus control healthy individuals. In the last video, we looked at two-dimensional gel electrophoresis coupled with mass spectrometry as a valuable tool for proteomics analyses. But despite the value of that technique, we also saw that there were some pitfalls and limitations to that. Namely, that if we have proteins that are not able to be fully separated by 2D gel electrophoresis, we won't be able to distinguish them in the comparison of treatments and controls. Likewise, we also saw that proteins of low abundance can be easily overshadowed by proteins of high abundance, because if the signals occur at very similar locations in the gel, the abundant protein is going to completely overshadow and eclipse the lower abundance protein, making that lower abundance protein difference between treatments and controls not detectable. Another issue with 2D gel electrophoresis and mass spectrometry is that the method is inherently not particularly quantitative. It's more of a qualitative method rather than a method that can statistically evaluate the absolute or relative difference between treatment and control samples. And what we are going to do in this video is talk about proteomics using isotope-coded affinity tags as a way to overcome some of those limitations of 2D electrophoresis plus mass spectrometry as our proteomics tool. So here we're looking at proteomics using isotope-coded affinity tags as a way to enable things like relative quantification of proteins between treatments and controls. That's something that 2D gel electrophoresis wasn't generally exceptional at, but isotope-coded affinity tags will allow us to do. Likewise, isotope-coded affinity tags in many cases will allow us to detect lower abundance proteins and compare those between treatment and control samples in a way that is superior to using 2D gel electrophoresis. So let's get started by talking about what we mean when we say an isotope-coded affinity tag. As is the case in any type of omics comparison, we will have a collection of molecules that we want to evaluate how they differ between treatments and controls. In the case of these proteomics experiments, they will of course be protein mixtures. So I'm going to put T for our treatment mixture of proteins, C for our control. This could be, for example, diseased individuals versus control individuals that are healthy, and we want to determine biomarkers at the level of proteins to distinguish between these samples to pinpoint what proteins are perhaps involved or perturbed by the disease state. So we generate these protein mixtures, extract those from whatever cell type or tissue type that we are looking at, and then what we will be doing is covalently tethering either the full intact proteins from the treatment versus the control to an isotope labeled tag, or we will first digest each of these protein mixtures using proteases such as trypsin that we talked about in the last video in order to create peptides from these simpler fragments. So what I'm going to put here is that optional as the first step in this is protease digestion. This is done for a few reasons, one of which is that, as we talked about in the last video, the process of mass spectrometry is generally simplified and more robust if we are using peptides rather than full, complete, intact proteins. And so the protease digestion simplifies the mixtures a bit in terms of their capacity for mass spectrometry. And then from there, what will happen is that we will have either the complete proteins or peptides, depending upon whether or not the sample has been digested. So we'll have that for both our treatments and our controls. And then each of those will be subjected to derivatization with an isotope-coded affinity tag, meaning that we are going to be reacting the protein or the peptides from the treatments and controls with a specific type of tag. And that tag that we will react with has three components that you should be aware of. First, it's going to have what we refer to as the affinity portion of the molecule. The affinity portion is a portion of the molecule that will later on in this workflow facilitate us in separating the proteins or the peptides from other biomolecules that are present in the cell for analysis by mass spectrometry. So the affinity tag will help us simplify the mixture of different compounds and proteins, lipids, etc. from the cell at a later step. So we have our affinity tag. Additionally, we have what we would refer to as the mass-coded linker. And what's going to be special and unique about the mass-encoded linker is that it is going to have specific isotope-labeled atoms here. And those specific isotope-labeled atoms are going to differ between the treatment and the control sample. For example, the treatment might have some atoms in that linker that are enriched with carbon-13, whereas the control would instead have mass enrichment with carbon-12 atoms, hence allowing mass spectrometry later on in this pipeline to distinguish between the tag that goes with the treatment and the tag that goes with the control, because the tag going with the treatment would end up having a different molecular weight and hence a different molecular ion than the control, due to the fact that the treatment has a different carbon isotope than the control, or has a different hydrogen isotope than the control, has some difference in isotopes, leading to a difference in the mass to charge ratio of the ultimate molecular ion of those compounds. So this tag that we are going to react the protein or peptide with has the affinity portion of that molecule, a mass-encoded linker, which means that there are specific isotope-labeled atoms that will differ between what we react with the treatment versus the control, and then finally, a specific functional group at one terminal of this molecule that will react with certain functional groups of the protein or peptide. So I'm going to put reactive functional group here. And the purpose of that reactive functional group is going to be to enable covalent linkage with the protein or the peptide. So it's going to react with specific functional groups in the protein or peptide in order to covalently link this whole set of the affinity portion, the mass-coded linker, and that functional group to our protein or our peptide. And so for the treatment samples, be they peptides or proteins, depending upon whether they've been digested or not, they would all get the same mass-encoded linker, meaning that this is enriched specifically with atoms such as carbon-12 or hydrogen-2. On the other hand, the control samples would be reacted with the same affinity tag. In this case, though, the mass-encoded linker, rather than having a carbon-12 label, which we said was happening up top here, or a hydrogen-2 label, instead would have a different isotope or isotopes present. So this could be enriched, for example, with carbon-13 instead of carbon-12, or enriched with hydrogen-1 instead of hydrogen-2. And hence, the tags that we are using for the treatment versus the control would behave the same chemically, because different isotopes will behave approximately the same chemically, but they will have different mass to charge ratios when evaluating by mass spectrometry. And then we finally have that same reactive functional group that we had up top for our treatments. And that enables the linkage covalently of this tag to our protein or our peptide. So the reaction would take place to join covalently the protein or the peptide from the treatments and the controls to this isotope-coded affinity tag. And so what would result after this chemical reaction to covalently link these is we will end up with our treatment versus control, proteins or peptides, peptide if we've digested with a protease, protein if we have not. And those are going to have, as I will put in blue here, a covalent bond linking them to the reactive functional group, because that's going to be the site that forms the covalent bond to the protein or the peptide. And that functional group is linked to the mass encoded linker. And then that is linked to the so-called affinity tag, which will facilitate us later on in purifying, separating these tagged proteins from ones that are not tagged or from other biomolecules within the cell. Same thing for the peptide mixture. It's going to form a covalent link via specific reactive functional group of the tag so that it's linked to the protein. And then for the control, same situation here where we're going to have a covalent link between the protein with the reactive functional group and so on down the chain there, or the peptide with that reactive functional group and so on down the chain. The difference between the treatment and the control will be that the mass encoded linker will differ between the two. So let's say that the mass encoded linker for the treatment is enriched with carbon-13. Mass encoded linker for the control is enriched with carbon-12, we'll say, in this case. And those could be whichever you want there, however you've designed the experiment. So we could say that we have carbon-12 up top here as our linker for the treatment and carbon-13 for our control. As long as we've kept track of which one has which, it will be fine because carbon-12 reacts equivalently to carbon-13 in these experiments. And so your treatment and control sample will differ in terms of their isotope label, and that will enable us to distinguish between proteins that originated from the treatment versus those that originated from the control. So after we have tagged our proteins here with these different isotope labeled tags, what we can do is mix the proteins from the treatment and control sample, because now we will be able to distinguish between them due to the fact that the treatment samples have a carbon-12 mass-coded link. The control samples, on the other hand, have a carbon-13 mass-coded link there. So we mix the two together, and then to further clean up the sample before mass spectrometry, some sort of affinity column would be used. The affinity column is a column chromatography packing that will enable the stationary phase, the chromatography packing, to selectively bind tightly with the proteins or peptides that have the affinity tag on them. And so hence, through that affinity column, all of the biomolecules, such as untagged proteins, lipids, nucleic acids, and so on, will flow right through the column very quickly, except for the proteins or peptides that have the affinity tag. Those with the affinity tag will more strongly adsorb to that stationary phase of the column, and eventually, using specific buffers, will be eluted from the column, meaning will be released from the column. And so as a consequence of that, you'll end up with a mixture of nearly exclusively, I say nearly exclusively, not exclusively, because no process is 100% efficient and accurate. So mixture of nearly exclusively the affinity labeled proteins or peptides. And then at that point, the mixture has been simplified enough by virtue of separating away and discarding all the non-affinity labeled molecules so that this can be subject to mass spectrometry. And with mass spectrometry then, the resulting mass spectrum that will result from this, if we look at the mass to charge ratio, so I'm going to make a mass to charge ratio plot here, m/z, mass to charge, and then relative abundance here on the y-axis. We normalize that relative abundance to 100. And if we take a look at the mass spectrum, and let's say we have signals show up here at 550 and 560. The difference there in the mass to charge ratio between these two is a difference of 10. And now that difference of 10 is important and relevant because if our label for our mass-coded linkage had 10 carbon atoms in it, and one of those was labeled with carbon 12 and the other with carbon 13, meaning that the label up top here had 10 carbon 12 atoms, the label down below here had 10 carbon 13 atoms, that would mean that the mass to charge ratio difference between the treatment and control would be equal to 10, since each of the individual carbon atoms of those 10 carbons had a 1 mass unit difference between carbon 12 and carbon 13. So that equates to a difference of 10 here. And so what we can do is look at the mass spectrum and compare the integrals, the intensities, for the signal right here that would correspond to the treatment versus the signal here that corresponded to the control. So this would be our treatment signal, and this is the control signal. We know which one is which because if we go back up to the top here in our hypothetical example, we said that we had a carbon 12 label on the treatment, a carbon 13 label on the control. So therefore the control is going to be the more massive of the two when we look at the signal to noise ratio of those. The treatment on the other hand is going to be the lower mass to charge ratio of those two. And we know these two are linked because of the fact that that mass to charge ratio difference is 10, which 10 corresponded to the number of carbon atoms that we hypothetically assumed were in this linker. So if our linker has 10 carbon 13 atoms in the control, which I'll put in C, versus 10 carbon 12 atoms in the treatment, that's going to result in this sort of mass to charge ratio difference. Now keep in mind there's a variety of different linkers that can be used here and a variety of different isotopes that can be incorporated there, and that would result in a different mass to charge ratio spread. But what we can do is, since these treatment and control samples were run in the same mass spectrum, the same experiment at exactly the same time, that means that it's apples to apples in comparing the integrals of these two signals. And so to get relative quantification of the amount of that particular peptide or that particular protein in the treatment versus the control, we can obtain by comparing the integrals of the signals for the treatment versus the control. So to relatively quantitate treatment versus control, compare the integral for the signal that we know corresponds to the treatment versus the one we know corresponds to the control. And that will tell us by what factor has the protein been upregulated or downregulated in the treatment relative to the control, without incorporating additional internal standards where we know exactly on a mass basis how much of that particular protein or that particular peptide there was. We wouldn't be able to get absolute quantification here, but we can definitely get relative quantification because we are running these two simultaneously. And when we're running these simultaneously, we can compare them side by side since they have the exact same composition except for what isotope is present there. And we assume if they have the same composition except for the isotope, they're going to ionize with the same efficiency and have the same properties and detectability by mass spec, so that we can do here a side-by-side comparison of the integrals for each. On the other hand, if we had ran these as two separate mass spectrum without mixing the samples beforehand, when we mixed them up here, instead we would not be able to do such a rigorous quantification comparison of these two, because there can be variability from one injection to the next when running mass spectra experiments. And so it's really advantageous and key to the quantitative capabilities of this method that the treatment and control sample are mixed during most of this process of preparation through affinity chromatography and handling, and then mass spectra, because if there's any variability in the method here at any of these steps from the time that the sample is mixed, the variability would affect equally the treatment and the control sample, meaning that if a bit of the sample was lost because it got stuck in a pipette tip, you would be losing the same amount of treatment sample as control sample because of the fact that those are already mixed together, and hence the relative amounts of the two proteins or two peptides would remain constant at that relative basis. And so this allows us to relatively quantify the treatment versus the control, and that is super valuable in terms of thinking about some of the fine scale changes that take place when a particular organism is in a disease state, or we're trying to determine the effect of UV radiation, or whatever other variable we want to explore the impact of the proteome on. So we've thus far rather generically looked at this isotope labeled or isotope coded affinity tag by just giving some generic terms for the affinity tag, the mass coded link, and the reactive functional group. Let's look at an example of a very common isotope coded affinity tag. So in this, an example of an isotope coded affinity tag. Isotope encoded because of the fact that we have that linker portion that has a different isotope present in the treatment versus the control. Affinity because we are going to include an affinity label that will allow us to separate out proteins or peptides that have been tagged versus biomolecules that have not been tagged. And then isotope coded affinity tag because of the fact that we have that tag on there. So an example of an isotope coded affinity tag, starting with the affinity tag portion of this molecule. Very commonly, biotin, the biomolecule that I'm drawing out here, is used as an affinity tag that will enable proteins or peptides to bind, meaning interact strongly with the packing of the protein chromatography column. So if we go ahead and draw out here this biotin affinity tag, I'm drawing the structure of biotin right here. Our biotin, which I will go ahead and label this portion of the molecule. This is our biotin affinity tag. Biotin is commonly used as an affinity tag because the biotin will strongly interact with and adsorb, it will strongly bind with the avidin as the stationary phase when we do chromatography with this. Stationary phase is the packing material of the chromatography column. So in other words, once we have this biotin covalently linked to our protein or peptide, the protein or peptide will very strongly interact with the avidin that is packing the chromatography column and remain adsorbed to the chromatography column as the non-labeled peptides, non-labeled proteins, and other biomolecules come right off. And so this helps us down the pipeline here in separating the isotope labeled proteins from other molecules that are not isotope labeled to simplify the mixture for the mass spectrometry process, since it is not generally possible to look at a complete crude mixture of different compounds by mass spectrometry because the mass spectra become too complex and the presence of other compounds can interfere with the ability to detect compounds of interest. So we have our biotin tag, and then the next portion of this molecule is that we link the biotin tag via an amide linkage here with what we refer to as the mass-encoded linker portion of our tag here. So what I'm going to do is just draw out an example of a common linker. This common linker, you will notice, has some ether groups. That is no coincidence that it has some ether groups. Ether groups are generally relatively non-reactive functional groups. If you think back to organic chemistry, there weren't very many reactions that were learned regarding ethers, and so they are commonly incorporated in these sort of non-reactive portions of the molecule. And so we refer to this part that's in blue here as our mass-encoded linker. And this is the portion of the isotope-encoded affinity tag that is going to differ between the treatment and the control. And so within this mass-encoded linker, what will happen is that the mass-encoded linker will have either some or all of the atoms, the carbons, that will be carbon 13 versus carbon 12 for the treatments or the controls, or alternatively, hydrogen 1 versus hydrogen 2 is another common way to go about this. And so in the case of a hydrogen 1 versus hydrogen 2 linker, we could say at this position, these X atoms here represent hydrogen 1 for all of the treatments, hydrogen 2 for the control at those positions. And then we could do similarly over here at this position, putting in these X atoms. And so as a result of this, what you will notice is that there are a grand total of 8 X atoms in here, where X is equal to hydrogen 1 for a treatment, hydrogen 2 for a control. And so what that means, we can interpret out of this, is that since there are 8 hydrogens that are going to be isotope labeled, then we would expect there to be a mass to charge ratio difference of 8 between the treatment and the control, where if we say that the control had hydrogen 2, the treatment had hydrogen 1, then it would be the control that would show up as the more massive signal, the larger mass to charge ratio signal in our mass spectrometry experiment here. But we're not done yet with this isotope-encoded affinity tag, because in addition to having this isotope labeled business and the portion that enables the molecule to bind to a specific stationary phase in chromatography, we also need some portion of this tag that will enable it to form a covalent bond to the protein or peptide so that we have this group linked onto our protein or peptide for analysis. And so for that portion of the structure, in this example of an isotope-coded affinity tag, we have a reactive group, a group that will react with specific functional groups from the protein. And in the case of this, what we have here, you will notice at the terminal end of this is an iodine atom. Iodine is a good leaving group in nucleophilic substitution reactions, and a sulfur atom of the thiol functional group in cysteine will readily react with the carbon atom right here in a nucleophilic substitution reaction, causing the iodine leaving group to leave. Hence, if we envision that we have a cysteine residue, which I'm just going to abbreviate as our SH group with a squiggly line here representing the rest of that cysteine amino acid residue, which could be present as part of a full intact protein or present of a peptide, what will happen is that the sulfur atom here is going to act as the nucleophile, comes in, forms a covalent bond to the carbon there as the leaving group leaves in an SN2 type reaction. And so as a result of that, what we will end up with on the product side of this equation, once the reaction has taken place to link our tag covalently to our cysteine, is that you will have, starting with, just to show part of this molecule, the rest of it is here represented as a squiggly line, but we'll start with the NH group here that is linked to our carbonyl, and that's amide linkage. And then we have our carbon that was bonded to the iodine leaving group that will end up ultimately bonded to our sulfur atom. And the sulfur atom then is bonded to the rest of the protein or the rest of the peptide, depending upon whether we're looking at the full intact protein at this point or the peptide at that point. And so that will give us then our protein or our peptide that has covalently linked to the mass encoded linker that differs between the treatment and control, so that we can distinguish between treatment and control by mass spectrometry, and additionally has this affinity tag attached covalently so that we can readily use chromatography with specialized stationary phases such as the avidin stationary phase to enable us to readily separate these tagged peptides or proteins from biomolecules that are not tagged, creating a really valuable way to carry out comparisons of the relative quantities of individual proteins and peptides between treatment and control samples. And this makes this isotope encoded affinity tag method a super valuable and one of the best approaches out there to relative quantitation-based proteomics and comparative proteomics, where we're comparing side by side treatment and control samples, because this will allow us to detect many of the proteins that would be missed using the previously discussed two-dimensional gel electrophoresis with mass spectrometry approach. The downfall of this is that it does require the use of these isotope encoded linkers and specific functional groups at the end here in green, which will react with specific targeted functional groups of your protein. So in some cases, some proteins could be overlooked if they don't have the correct functional group to react with the isotope encoded affinity tag. For example, in the case of this tag, a limitation would be that this functional group specifically reacts with the thiol group of cysteine residues. And so if you're working with a protein that doesn't have any cysteine residues, then that protein would be completely overlooked because there are no cysteines to react with the reactive handle of our isotope encoded tag. So no one proteomics technique is absolutely perfect in terms of its ability to detect all of the proteins within a sample, but generally speaking, these isotope encoded affinity tags will allow us to detect a wide range of different proteins or different peptides and enable them to do it in a way that is suitable for determining the relative quantities of material of specific protein between treatments and controls.